Hello, and the topic for this video is going to be DNA replication. We're going to go over some of the basics in this video, and then I've got a second video for you that'll show a little more detail of what's happening at the replication fork during DNA replication. One of the key things about DNA replication in eukaryotes is that DNA replication is semi-conservative. Okay? What that means is before the cell undergoes DNA replication, all right, we may have a matchup something like this with our base pairs, right? And we know from complementary and base pairing, remember A is always going to pair with T, C is always going to pair with G. So if you have one strand of DNA, you can always figure out what the sequence or the order of the nucleotides is on the other strand, okay? So in pink here, this is what we're going to call our parental strand, okay? Now that strand is going to run, or that DNA is going to have a directionality to it. It's going to have a 5' end and a 3' end. The other side is going to be anti-parallel, okay? So the two, oops, that's not right on spelling. That's better, all right? So the two strands in that case are what's known as anti-parallel. So you have one strand that runs in one direction, the other strand goes in the other direction. Now remember that 5' and that 3' designation gets to the carbons on the sugar. The 5' end is going to be where the phosphate is going to be. The 3' end is where a new nucleotide can be added, okay? So we've got our parental strands. What happens with DNA replication is our two strands are going to separate, okay? So we've got the one A, C, G, T here. And then we've got our other strand that we'll put over here that runs the opposite direction. It's going to be A, C, G, and T, okay? What's going to happen? There are going to be a number of enzymes that are going to be a part of this process. And I'll talk a little bit more in detail about those enzymes in the next video. But I'm going to mention some of them here, just so that you hear it more than once. And I'll write out the whole name. We've got DNA polymerase is going to be one of our main enzymes. That DNA polymerase, I kind of call that the photocopying enzyme. That's the enzyme that's going to make our new strand of DNA. Okay? What we're going to have happen there, that DNA polymerase is going to come in and it's going to fill in a new strand. And I'm going to use green here so you can see the difference between our pink and our old strand and our new strand. Okay? So our new strand is going to be in green. Okay? And it's going to go opposite of what we've got here. So we would fill in A, C, G, T here. On this side, we're going to fill in A, C, G, T. Okay? Then what's going to happen? All right? We now have two copies of that DNA, all right? And it's semi-conservative because you have one parental strand and one strand that's newly synthesized. That's what it means for it to be semi-conservative, okay? Now, in terms of DNA replication, when this is going to occur in a cell, typically is going to be just prior to the process of mitosis. And we haven't talked specifically about mitosis yet, so that mitosis is the process of making new cells or cell division, okay? So DNA replication happens in the cell. It's going to happen in the nucleus because that's where the DNA is contained. Happens just prior to mitosis. We start out with a parental strand. We end up with 2 strands of DNA, 2 identical copies. Both have 1 parental strand and 1 new synthesis, okay? So we've got that process there, just kind of briefly what DNA replication is. I want to kind of add on to some of the enzymes here. DNA polymerase is one that I've talked about, all right? One of the key things about DNA polymerase, and this will become a little more clear, I hope, in the second video when I show you what's happening specifically at the replication fork, okay? DNA polymerase only moves in one direction, okay? And that direction for DNA polymerase is 5' to 3', okay? So that's the direction that that particular enzyme has to move on the DNA template, all right? The DNA template is going to be our parental strand, okay? That DNA polymerase is going to read what's in the parental strand, fill in the corresponding nucleotides, all right? This creates a little bit of trouble with DNA replication that I'm going to talk about in the next video, all right? But just be aware, DNA polymerase happens in one direction, all right? We're going to have some other enzymes that are going to be important. We are going to have an enzyme called helicase, all right? That helicase is what opens up our double-stranded DNA. So in my short schematic over here, what we have to do with that parental strand is we have to pull them apart. We have to separate them. And it's going to be DNA helicase that's going to separate those strands of DNA so that we can start the process of DNA replication, okay? There's another enzyme called topoisomerase that's going to be important as well, all right? Topoisomerase is going to prevent essentially twisting of the DNA molecule. Because it's arranged in a double helix and that's kind of spiral in shape, when you pull apart those 2 strands of DNA with DNA helicase, that puts some stress on the DNA, and you don't want to break apart the DNA. So the topoisomerase comes in and relieves that stress so that the DNA molecule doesn't break apart. Okay, kind of in a nutshell, that's what's happening. Okay. The other thing we're going to have involved in DNA replication, we're going to have a RNA in this case. Make sure you get that right. It's RNA. I wrote it correctly. Okay. RNA primase. And we need that RNA primase because DNA polymerase can't just attach to a naked strand of DNA. It needs something to attach to. And that thing that it attaches to is a little bit of RNA that RNA primase puts down so that DNA polymerase knows, okay, this is where I need to come in and start making new DNA. Okay? So that RNA primase puts down or adds a short, okay? It's a couple base pairs, maybe 4 or 5 base pairs. I'm not going to ask you to tell me how many base pairs it's putting down. I just want you to know it puts down a short sequence of RNA, and it does that so DNA polymerase can start DNA replication. Okay? So we got DNA polymerase, helicase, topoisomerase, RNA primase. We are also going to have DNA ligase. That's kind of related to our RNA primase because what we're going to have, you'll see in the next video, there'll be some gaps in the sugar phosphate backbone. The DNA ligase is going to fill those gaps in our sugar, I think you can see that, yeah, phosphate backbone. Okay. There are going to be a couple more enzymes and proteins that are going to be involved in DNA replication, but I've kind of run out of room here, so I'm going to introduce those in the next video. I'll also, again, remind you about each of our enzymes here that are going to be important. Now, what I want you to keep in mind is the list that I give you for enzymes are enzymes that are important in DNA replication, but it is not a complete list of all the enzymes involved in DNA replication. These are the ones I want you to be able to know and know what they do and know where they are on the DNA replication fork. Okay? Thank you for your attention. If you have any questions, please let me know.